now is Mr. Scott Drouin, PhD. He's a senior consultant, toxicology and food safety for the Rimkus Consulting Group. Scott has over 23 years of toxicology experience. He's an expert on health risk from substance exposures in food, and he's trained as a National Institute of Allergies. Let's see if I get that right. Allergy and Infectious Disease. Scott, did I get that right?
You did. Thank you very much, Stephen.
Infectious Disease fellow. Listen, being a fellow is a big, big deal, ladies and gentlemen, just so you know, in human infection and disease. Scott's going to talk to us today, excuse my voice is going already, on knowing what's in your food, a toxicological analysis of food ingredients and contaminants. So important with what's going on today. Take it away, Scott.
Okay, next slide, please. Thank you, Stephen. I appreciate the introduction. As a person that studies respiratory biology, lung biology, I also appreciate that breath before the talk, get all that oxygen and so forth. I burned a lot of calories just listening to Dale's talk, so this is a good thing. So this morning, welcome everybody. Thank you for attending my TED Talk. I'm going to be discussing this topic, knowing what's in your food, a toxicological analysis of food ingredients and contaminants. Now, I'm a trained toxicologist, and I have strengths in immunology and, as I said, respiratory biology. And with this comes 24 years working in a lab or around lab environment. So a lot of my background going into this particular area involves testing expertise. You know, how do we create an experiment to test a question, test a hypothesis, that's the word that we use in the field, or a premise, to have an evidence-based answer. And when we enter various legal matters or insurance claims where we are asked, based on our experience, our expertise, to answer a question, it's because we know the skills and methodologies to come up with a testable hypothesis and come up with a meaningful answer. And so this is really going to be the gist of my talk. Next slide, please. Okay, so I've introduced this before for other talks, food safety, or what else is in that food you're consuming. So the issue here that we're faced with is in the past 2 years with COVID, this is a way that everybody's familiar with, people have become very, very sensitive and very conscientious of contamination. Now, in the context of COVID, it is contamination in the air, something that you breathe. Now, I can always come back for future talk on COVID, but today I'm going to talk about food safety. And food safety is a major worldwide concern because a variety of chemicals, microorganisms can contaminate food, and they can exert health effects. Now, the sources of these contaminations are quite varied. They can be either natural, such as bacteria, or they can be human, like pesticides. These are examples. And they exert problems in the hospitality industry because basically nobody wants to get sick. There is an inherent trust when you go into a restaurant, and whether you're getting a glass of water or you're ordering some eggs or seafood, you expect that food to not cause you illness. Whether it's contaminated or not is a subject for debate, but not cause you illness. And so therefore, there are mechanisms in place to prevent this, and they could be sort of quality assurance, SOPs. These are methodologies that mitigate the risk of a food becoming contaminated, and so the food is safe to eat. Now, what happens when that breaks down? Well, that's where I step in. Next slide, please. So basically, at Rimkus, we within our day-to-day operations, with the projects we come in, we'll have either legal matters or insurance claims come in where there is some allegation of food contamination. And either an attorney or adjuster who represents typically a restaurant or it could be some sort of food manufacturer or so forth. They'll come in with a problem, with an allegation of a food contamination, but they need to understand how this occurred. And that's when they approach us. And we basically, we take data that they provide us, and sometimes it's testable, sometimes it's a record review, but we we identify the problem and then come up with an answer based on evidence. So with the cases we come in, there's actually quite a range, believe it or not. So a lot of times with, on the left-hand side of the scale, there's a phrase, "I don't know what this is, can it hurt me?" So in this case, what happens is there is an observation in some sort of food process where they see a contamination or a potential contaminant. And since they don't know what it is, they don't know if it can hurt a person, or maybe they think it might hurt a person. So they will approach us to either to identify what that contaminant is, and two, conduct a risk assessment. So as toxicologists, we're very good at delving into literature and identifying if this particular item is a health hazard for a person or not. So these sorts of claims, the key feature is that this is before anybody gets hurt. And this is actually very important to decide, because then there are two things will happen. In an insurance claim, for example, they can claim this contaminated product as a loss, and they can prevent anybody from getting injured, which might foster lawsuits or other things down the line. So it's a kind of a preventative measure, and that's the role that we play in this scenario. In the middle scenario, it's like an object or some sort of contaminant is observed, and they say, "I don't know what this is," but they presuppose that it hurt me. So there is an association here between seeing something unknown in a food item, and then you have some sort of injury, and then drawing that association. So in this case, we will typically step in and again identify what that contaminant is. And as I will state in additional slides from here, we'll take steps through standard methodologies to identify if that object did cause this particular injury. And then the last one is, I know what this is, and it hurt me. So these are claims where people will state that, I see this object, I based on my experience, I can identify what it is, and because of this, I know. So it's an even stronger association between a subjective opinion about identifying an object and knowing how this is going to affect a person's health. And again, in this case scenario, we will take steps, usually through a lot of medical records, to kind of vet what the object is it what they say? So there's some sort of support to verify or validate this part of the claim, and then identify if it has an association with that injury or not. I would also add that as part of the spectrum of these contamination claims, I'm heavily involved with testing based on my background experience, but there's also sort of a food quality aspect to this as well. We have consultants, Jane Roth, a wonderful person that works with us, who specializes in evaluating standards of care, SOPs, in restaurants, food manufacturers, and so forth. And this is really an important resource for us, because it sometimes, if there is a contaminant identified, we need more information to understand where it originated. And so if you can have a person look through SOPs or food processes to kind of understand where this originated, it helps us better define our testing strategies, and also looking at the downstream health effects from that. So that's a whole other level I'm not going to talk about, but we have other people that can deal with that with these cases. Next slide, please. So the key thing to consider here is I'm a toxicologist first and foremost. And the thing that a toxicologist does is we have criteria to establish cause and effect. And a lot of our, when a client contacts me, I will go through this list. I won't ask them these terms specifically, although I like talking about science, so I may educate them a little bit about them. But I will ask them for records that will establish as much of these 5 points as possible. And attorneys tend to have a greater breadth of information that will support these different points. Adjusters don't always have it, so you got to make do with what you have. But we're resourceful. There are a lot of things we can figure out on our own. And so when you deal with cause and effect, you have to first assess exposure, especially with a health injury. Did an individual actually, were they exposed to a certain amount of substance that was encountered in the environment? So a common thing that most people hear about, especially in this day and age, is salmonella contamination. So they, in a particular scenario, somebody might go into a restaurant and then they eat a dish, perhaps it has eggs or chicken. They get sick with salmonella. They get sick a few hours later. Sorry about that, I'm jumping the gun here. They get sick a few hours later, maybe they have a fever, nausea, vomiting, and then they'll immediately associate that with the meal that they just had. Now, the question there is if you have that scenario, like I think I know what is in there, without any testing evidence, it's just a very subjective opinion. A lot of times what we require first is that a person will go into the hospital or clinical environment. They'll have usually some sample tested, blood or stool. And if it tests positive for salmonella, then you have part of the puzzle that you know that they are experiencing some sort of salmonella poisoning. And that sets up to a question is where do they get their exposure? And so on the flip side, we would start looking at we'd conduct a trace back and look at their food history, what they've consumed. Perhaps the restaurant has some sample of food remaining in a preserved state that can be tested for the presence of salmonella to kind of make that link. The other aspect of this is dose. So the issue with dose is that and this is in this day and age, this is missed. So there are a lot of things that people associate as being toxic, but it takes a certain amount of the substance to cause injury. So alcohol is a very, very good example of this. So a lot of people consume alcohol. They don't necessarily think of it as toxic to their bodies, but if they drink too much, it can be lethal. So this is a very good example of this this sort of methodology. Is it's not only establishing exposure, but if we can, is there can we identify the amount that they consumed, and then is that amount associated with a particular injury? Dosages are very difficult, and a lot of times this is something used a lot in risk assessment, where you can say, well, if we know this amount in the food, and it's going to be above this amount that people are going to be exposed to, then they're going to have a particular injury. But by and large, for a lot of the legal cases where you're looking at injury, dose will be difficult to establish, at least for the hospitality area that I'm talking about. Our alcohol analyses are actually quite good in terms of assessing dosage and so forth. But that methodology is very, very well characterized. Now, if we cannot come up with an exposure dose, this leads us to the third point. Can we come up with an alternative exposure? In the legal arena, alternative causation is a big thing. So if you cannot establish an exposure as part of the allegation, can you look at other records? And in this case, medical records are quite useful in the case of an injury. And is there some other type of exposure that would explain the health injury experienced by the claimant or the plaintiff? There are also confounding factors. These are items that might influence the exposure or influence the injury that would explain them as well. And then finally, one of the toxicologists' greatest tools is developing a timeline. In this case, especially with bacterial infections, there is a well-characterized background of medical literature that documents, based on large-scale epidemiological studies, when a person would experience symptoms after an exposure. And basically, you're not going to experience symptoms prior to the exposure. That goes against the laws of nature and time and so forth. That violates causation. If anybody's watched Star Trek will understand that. So the issue here is developing a timeline, and having all the information to develop a timeline is very, very important. And it's something I harp on a lot for not only hospitality and food safety cases, but a lot of the other medical cases that we deal with, a lot of mold cases. And so establishing when a person's exposed relative to their symptoms is a very important tool to help help establish cause and effect. Next slide, please. So obviously, I've talked a lot about our toolbox and what we look for in a general sense. In the next 3 slides, with our casework, I've developed these into 3 case studies that kind of exemplify how we use these tools. And if you remember the 2 slides back, I discussed these different scenarios about conducting risk assessments, looking at injuries, identifying the contaminant, and so forth. So here in these next 3 slides, I'll discuss 3 case studies that will help, hopefully help you understand how we use these tools. So about a year ago, I received a case that dealt with chemical contamination. And I'll never forget, the adjuster came in and I sat down and discussed the scope of her legal claim. And the question that was asked was, what are these floaties in my spring water? Are they hazardous? So that's quite a broad question. As a scientist, you know, that good scientific term, floaties, you know, what is that? But it was a legitimate concern. So in this claim, there was spring water bottled up in something that's supposed to be natural, healthy, not have any contaminants. And what happened is the people that were consuming this water noticed in the bottles that were shipped to them, they had all these floaties. And so there was a process where these bottles were shipped back to the vendor. And the claim was that this contamination appeared, you know, when the processes were evaluated, the contamination appeared after changing the soap vendor. So the claimant basically wanted to know what were these floaties? Were they something from the soap vendor? And are they hazardous? That was a big question. Nobody at that point had expressed any health injuries from consuming this contaminated water, but that was a big concern. So the first step after conducting this initial interview and developing the scope is they wanted testing done, and they wanted to identify what these floaties were. So we had bottles shipped off to a lab, and we have the capacity either internally or through third-party laboratories to look at either microscopically or at the molecular level, identify what these unknown contaminants can be. So in this particular case, the contaminant was actually identified as a bacterial energy storage chemical. And you know, this is a product that was quite common. It was a carbohydrate. You know, obviously bacteria, they, like any other organism, they need energy and they'll store it. You know, us as humans, we store it in fat. Well, these bacteria will store it as a carbohydrate moiety, and that's what these compounds were. And that was a concern because for both the insurance and the claimant, because the claimant was not protected for bacterial contamination, and so there was some pressure to kind of delve further in terms of where this originated, and you know, so that the result actually fell into this gray area. And so we did initial studying to kind of attempt to identify the source, and we eliminated the soap. So the one concern was that maybe the soap was contaminated with bacteria that might be the source of this energy molecule, but we went through this initial process to identify if it was bacteria or not. At the same time, I was delving into the literature for this molecule, and the thing is that in today's business environment, if you find a chemical that is useful as a tool for making other products, that is something a industrious company or an entrepreneurial company will do. And so what I found is that chemical was actually used in plastics, and because it's biodegradable. And I discovered this in terms of evaluating if this product was safe or not, and it was. It actually, this particular chemical was used in clinical devices as a plastic or polymer, because you can basically use it as suture material, especially for like heart surgeries and so forth, because over time it would biodegrade in the body, and it was considered quite safe. The other thing it was used for is in bottles. And so this item would be used in bottles because of it made the bottles more easy to recycle. So in evaluating this, I came to the conclusion that, or came to understand that this chemical is actually part of bottles, but it also, because it's biodegradable, it suffers from certain extremes, heat, low pHs, and so forth. What we realized is that about the time of the, there were two things that kind of supported this as the source of the contamination. So one, when we reviewed the food processing logs, there were issues with cleaning of the water bottles from this vendor, from the cleaning vendor, where they were using much higher temperatures in the bottles. And so this supports the background literature that basically this chemical will break down above this temperature threshold. The second thing was, if it was a bacterial contamination in the bottle, what was very important is that the contamination in the bottle itself was not uniform. Invariably, the contamination, the floaties, were all concentrated around the neck. Well, if you actually looked at the food process and cleaning of these bottles, you actually had really hot water spraying towards the neck in that area. And that's where a lot of the hot water would settle during the cleaning process. And so the argument here is that in these scenarios, you have hot water and the pattern of contamination coalesce with the cleaning process. And so in the end, it was deemed that this was the source and of this floating contamination. And basically you'd have the loss was legitimate. So moving on to the next slide. Another set of projects that we get quite often are foreign objects. And usually this results in somebody asking, what is this object? It broke my tooth. So the claim was, the claimant basically established that he observed a foreign object in his breakfast entree, and he claimed that it broke a claimant's tooth. So this project did not have as much information as the previous one, and literally all we had were a few photos and a set of dental records after the tooth. And so the adjuster, our client, basically wanted us to identify what this object was, and we did. It looked like a little piece of tubing, but basically we found out that it was a bread clip, and it was used to seal a bread bag. And the nature of its curling would indicate that it had been exposed to heat. The other testing that is very important is we tested it for human saliva, because there are allegations out there where somebody will say, well, it broke my tooth, but if there's no obvious evidence that they actually had the object in their mouth, then that would sort of abrogate the legitimacy of the claim. So we did test for human saliva, and it was detected on this object. Now, unfortunately, the dental records did indicate that there was a tooth injury, but records prior to this incident were not available to review. And the reason why that's important is because we don't know for certain. Now, this object could potentially chip a tooth if bitten at the right angle, and we did some research in the literature about that. But we don't know if that person had tooth injuries prior to this exposure to this foreign object. So, the other thing that would have been required too is we didn't have this information as well because the adjuster decided to close the project, the matter after this. But arguably the location of the bread supplies inside the restaurant would have been helpful to know, because if they're within vicinity of a heating source, like a frying pan, where the breakfast was made and so forth, given that this object was exposed to heat, would have been an issue. So that was a problem in terms of us having limited access to information, but we could actually make do with what we had in a rather nice way. Next slide, please. So the last case, oh, one more before that, please. So the last one is microbiological contamination. So we not only deal with food sources, we also do deal with cosmetics. And in this case, we had a salon that was administering a lip wax to a customer. And the customer came back and said, this lip wax caused my cold sore. And so they had some testing conducted, and the symptoms did appear after the application. And the testing that they had initially conducted were tested positive for HSV. So this really, this is a classic toxicology type case. So we both looked, we had enough records looking at exposure and at the injury that we can kind of come to an assessment here. So in terms of the testing, we actually looked at the testing records. And the problem with this approach was that there was only a serological testing methodology, and it didn't really establish when the person was infected. Now, with COVID, you have all these tests. You have PCR tests, you have antibody tests, serological tests. And serological tests will often tell you that you were exposed and you have antibodies present against the virus, but they don't tell you when you were exposed. Whereas a PCR test will tell you that you have an active infection at that moment. Well, in this case, they only had the serological test, so it did not establish the time frame for when they were infected. In addition, in terms of the exposure, we had records from the salon where it was obvious that the ingredients and the heating would actually render the HSV non-viable. And so in this case, if there was any contamination in the cosmetic product, the conditions would not favor its transmission. The final thing, which also would poke a hole in this allegation, was that and reduce or mitigate exposure, was that the employees would wear gloves when they were administering this cosmetic to their clients. So that also prevents transmission. Final slide, please. So in conclusion, in these studies, the successful analysis of food claims basically requires meaningful evidence. And we understand from a consulting business that we don't always have records, all the records we would like. But if you do approach us, be prepared for us to ask. So we will ask for records that deal with exposure, and this includes business records, testing results that document exposure to a contaminant. We'll ask for records that deal with timelines. So these are medical records both before and after the alleged exposure that document the appearance of symptoms and maybe established latency, which is very important for bacterial or microbiological infections that result from microbial contamination. And finally, the nature of the injuries, records that deal with this, and this would also be medical records. Are the medical diagnoses and testing results consistent with symptoms associated with the exposure? I've gotten cases where some people make outlandish claims, you know, this exposure caused my autoimmune disease and so forth. And we basically look through the literature and we pair out if that's a legitimate association or not. Next slide, please. So thank you for your time. Please feel to ask me any sorts of questions on this. I'm happy to help.
Scott, thank you very much. Interesting, in-depth analysis, fascinating case studies. Thanks for joining us. Scott, I'm going to ask you to hang around during the break, if you will, in the interest of time, so that if people do have questions, they can put them in the chat for you during the break time, if that's okay with you.
That's fine.